Good afternoon and welcome to this lunchtime seminar series organised through the NIHR Oxford Biomedical Research Centre Statistics Hub. My name is Ioana Marian and I'm a senior statistician at Oxford Clinical Trials Research Unit, NDORMS. Today I'd like to talk on what's so great about randomised controlled trials. The session will cover how we define randomised controlled trials, which is a refresher on a previous session from the series called Introduction in Clinical Trials. We'll then look at different types of bias which can arise in trial design and consider a key feature of this type of studies, which is randomisation. And we'll end with a summary. Let's look at a couple of example trials conducted here in our research unit. In the UK STAR study, patients with Achilles tendon rupture were treated with either a functional brace or a cast, and their outcomes compared at various time points in the study. By comparing two treatment options, the research team looked to answer the question: what is the effectiveness and cost-effectiveness of cast versus functional brace for non-surgical treatment of Achilles tendon rupture? In another study, the Seesaw study, the research team assessed 3 medical interventions in patients with subacromial pain and answered the question, what is the more superior intervention for subacromial shoulder pain between arthroscopic subacromial decompression, arthroscopy only, or providing no treatment? In each study had clearly defined hypothesis and involved some level of comparisons of treatment among different patient groups. So this might be a good refresher already just by seeing the example of what we mean by an RCT. When it comes to comparing medical treatments, randomized controlled trials are often referred to as the gold standard design and often referred to as a prospective study that compares the effect and value of one or multiple interventions against a control in human being. And note that an RCT is prospective rather than retrospective, so that study participants must be followed forward in time, unlike a case control, for example, where the researcher collects information retrospectively. So in a trial, participants are usually followed from a well-defined time point. This is usually called time zero baseline. They can be conducted to evaluate any different types of interventions or potential treatments like drugs, surgical procedures, physiotherapy, acupuncture, health education. Another way to look at an RCT is as a quantitative, comparative, a controlled experiment in which a group of investigators study 2 or more interventions, and there's a bit of an overlap, in a series of participants who are randomly allocated to each intervention group. And the key word to focus on here is the unique feature. It's a random allocation. It's a unique feature of this type of design for the aspect under evaluation. Let's try and take a closer look. In a very simple example of an RCT, we sample from a well-defined population, which we often refer to as the inclusion criteria, and then we randomly allocate patients to intervention groups, a bit like throwing a die. We then administer treatment and follow up participants to collect the outcomes that we're interested in. Now, the key element here is the randomization, and the randomization ensures that the groups being compared are on average similar, or they're balanced at the start of the trial for what is known and unknown risk factors. So these could be observed or unobserved characteristics of the patients. Because groups only differ with respect to their treatments and not any other factors, we can have a fair comparison of treatments. So down along the line, if we observe differences in outcome, we can infer this is because of the treatment that was received rather than any other factors. So there's a cause and effect relationship between the treatment and the outcomes. And this is, again, slightly different from an observational setting where a clinician might choose to give a patient a particular treatment So they might give a new treatment to sicker patients for whom they are more sure that standard care won't work. So where there's no treatment preference, or what we refer to as equipoise, it can be argued that from an ethical point of view, every participant is guaranteed a random chance of receiving the best treatment when they take part in a clinical trial. What is great about an RCT is that it allows us to compare the treatment effect in groups of participants who differ only with respect to their treatment. So we may observe an effect for several reasons. For example, in a nutritional experiment, it is possible that simply being in the study will make participants more careful about what they eat, also known as the Horton effect. We may encounter regression to the mean. For example, if we select participants because they have high or low values of some sort, for example BMI, we would expect that on remeasurement, their group mean will be closer to the mean for the whole population that we've sampled from. Participants may also have a placebo response just to the fact that they are being treated, or an actual biological response to the treatment. So when compared to the control group, we may observe very similar, we might call illusionary effects. But the real difference is what the RCT enables us to confidently focus on. So the true effects, and to remove the other factors that might mislead the interpretation of our results. So the key point is for an RCT is that we are comparing groups of subjects who differ only with respect to the treatment, and that's why we randomize. And when we observe that the outcome differs, it's reasonable to assume that it's because of actual true differences in the treatment. So that cause and effect relationship is clean as such. But a key condition is that the trial is performed well. One way to ensure that the trial is conducted well is through transparent reporting. And to facilitate this, the Equator Network has been developing several reporting guidelines, including the SPIRIT guidelines for how to write a protocol for a trial, the CONSORT guideline for what to include when reporting the results of a trial. Note that there is an extension for CONSORT due in 2025. There are various extensions depending on the trial design. However, transparency is only one aspect. So studies can be performed badly but reported well. So for that reason, we need to think about bias in randomized controlled trials as one of the main concerns, and something that can be caused consciously or unconsciously. We refer to bias as any systematic error introduced into the study at any stage, could be at the design, the execution of the trial, as we analyze or when we interpret the results. So the key question to ask is, does our trial give an unbiased answer to the research question? And for this, we need to look at what is an internal validity, meaning how well does our trial answer the question that it's set out to answer, and an external validity. How well does our trial results apply to the population we want to give the treatment to later on in routine practice? So is what we observe in a sample of trial participants true for the whole population? And the focus is to design and minimize this bias. Bias can enter the study at different stages. So from the way we design our research questions, how we recruit participants and randomize them into the study, or even how we report our findings at the end. Is the trial designed to favor a particular outcome, or where and how might bias or faults be introduced into a study design? These are the kind of questions to think about. Different types of bias can occur in trial design. Selection bias, which is a systematic difference in comparison groups. For example, would younger participants respond differently to older participants in wrist fracture treatment? We could observe performance bias, which is about systematic differences in the care provided across sites outside the intervention protocol. So there may be varying levels of consistency across participants in the same group. Attrition bias is a systematic difference in withdrawals or in participants who stay may differ from the ones who leave the study. So we need to ask whether there's any withdrawal imbalance. Participants less likely to respond to treatment, for example, may be more likely to drop out of the study, or the opposite because they're feeling so much better. So we can artificially favour the outcome in the intervention group. Detection bias can occur if there are systematic differences in outcome assessed or in the assessment of outcomes. For example, are participants in the treatment group assessed more thoroughly than in the control, and therefore we may see a positive result in the intervention. And finally, reporting bias, which would be a selective outcome reporting. So perhaps we might choose to report some interesting results, but not all the results. And this can be quite difficult to detect. Depending on the type of bias, we might consider different strategies to reduce the potential sources of bias. So for example, to avoid selection bias, we might consider randomization as a technique by using concealment, meaning that those who admit participants into a trial are shielded from knowing future assignments. The decision to accept or reject a patient must be made and informed consent obtained without the knowledge of the treatment being assigned. Now, to address performance bias, it may be that we decide to blind participants or clinicians and standardize care or the site's training. In attrition bias, we might choose to use an outcome which is likely to be well completed, or anything that we can implement to minimize loss to follow-up, perhaps monitoring, communication, incentives. To address detection bias, we could consider assessor blinding, so objective outcome measures, standardized protocols, and make sure that the same assessment method or frequency takes place across both groups. As for reporting bias, one way to tackle is by pre-specifying the analysis or outcomes in the protocol or a statistical analysis plan and make it publicly available. The most significant difference between clinical trials and other forms of investigation is randomization, and this is how patients are allocated their treatment as they join the study. What do we mean by random allocation? Well, it's one that's not an allocation that is not determined by investigators, clinicians, participants, or anyone else. It is an allocation to each group by random chance, and each participant has a known chance, usually an equal chance, of receiving each treatment, something that we refer to as one-to-one randomization. Treatment received, however, cannot be predicted, and this is what we mean when we say concealment. And as a result, random allocation is likely to produce very similar groups at the start, as we mentioned, those observed and unobserved characteristics. How do we ensure that a random allocation is successful? Well, this depends on two key factors. First of all is the actual generation of a random sequence, and secondly, concealing that allocation sequence. So before any treatment is given to the participants in the study, we have to choose how to randomize, and strategies for generating a random sequence can vary in complexity. So let's have a look at some randomization methods and how we can implement them. First, one of the most straightforward approach is simple randomization. This can be thought of as an equivalent of tossing a coin or rolling a die for each allocation. In practice, a randomization list is usually prepared by the statistician in advance or by employing a secure software. Here we have two treatments being randomized, treatment A and treatment B. At the end of our randomization, the total in group A should be similar to the total in group B. Now, this may depend on how many participants we have randomized overall into our study. We can also use blocks with our simple randomization, which can help to restrict the A-B balance within a set and ultimately ensure a balance overall across the study. Here we have an example of block sizes of 4, and if a block size is fixed and known, it can lead to predictability of allocation. But to prevent this, trials often use variable block sizes of different permutations. So for example, instead of using just a block of 4, they may use a block of 2, block of 4, a block of 6, and conceal what these are. These blocks may be used variably, so they're permuted blocks. A different randomization method is stratification. This can limit or restrict the A-B group imbalance on important prognostic factors for the study outcome. So for example, we might choose to stratify by disease severity because we expect a worse severity at baseline to impact a later post-treatment score. And what about if we wanted to ensure balance on sex and age, for example, under and over 50? Let's look at an example. Imagine that we're now randomizing participants into the study using the stratified randomization. The first participant to be randomized is over 50 years, they are male, and therefore they receive the first position, which is group A. A second participant, which is over 50, gets randomized. They are female, and their allocation is group A, and that's because group B has already been allocated. For each strata, we have a separate randomization list, and usually it's made of permuted blocks of variable block sizes. With already 2 factors and 2 levels, we have 4 lists. So quite quickly, the number of separate lists required gets out of control. And in this case, we run the risk that many lists have an uncompleted block, leading to an overall imbalance. So to know that this type of randomization is only feasible for a limited amount of stratification factors and levels. A different method is a dynamic method of treatment allocation called minimization. And this is where the allocation is based on participant characteristics, somewhat similar to stratification. However, it additionally accounts for previous allocations in order to minimize the treatment imbalance. So after the first randomization, there is a higher chance of treatment balance among participants with different characteristics at any point in the trial. In addition to generating the treatment allocation lists that we've reviewed, there are other practical aspects in randomization that need to be considered. Simply asking the question, when will the participant be randomized? Will this be on the day they are screened for recruitment or for practical reasons at a later stage? And there are different ways to deliver the allocation, some which are encouraged and others less. So the ones that use a less random approach, for example, deciding to assign group A on an odd date and group B on an even date, or depending on the date of birth of the participant or the date of the appointment, an approach like this can very easily introduce bias as they're not adequately random. Instead, a pre-prepared sequence with each allocation in a separate envelope may be a better idea, assuming the envelopes are opaque sealed, or setting up an office to call for the next allocation. In practice, we do see a lot more the use of secure internet platform that facilitates randomization. To summarize, a randomized controlled trial which is well designed, planned, executed, and reported can be a powerful experimental technique to assess if an intervention is effective. Bias can compromise a study, whether it's intentional, unintentional, conscious or unconscious, and it should be addressed from the design stage. And randomization is what sets clinical trials apart. Varying randomization methods can help achieve a good allocation balance, and their implementation should be delivered appropriately. Final remark is that clinical trials are only as strong as the weakest elements of design, execution, and analysis. A list of references, and I'm welcome to take any questions. Thank you for your time.